Hi, my name is Dr. Sharon Duerkop, and I work in the Department of Immunology and Microbiology here at the University of Colorado Anschutz Medical Campus. When I'm not in the lab, I'm usually taking care of my family and my expanding family. And I also like to read, and I enjoy playing soccer as well. So the research that our lab focuses on is how the body fends off different microorganisms like bacteria and viruses. And I specifically work on viruses. And I want to know how these two proteins that we study, NOD1 and NOD2, are involved in influenza virus infection. These guys specifically recognize different changes within the cell that happen. So they were originally identified to recognize parts of bacteria. And since then, we now know that they can recognize things like viruses, and they can also respond to different stresses in the cell. And then what they do is they end up causing a response, and then you have inflammation happen. And ultimately, inflammation will bring in different cells, immune cells. And these cells are important for controlling infection. So why are we interested in influenza virus? As you all know, influenza virus, also known as the flu, is a pretty bad, pretty nasty virus. It is estimated by the World Health Organization to cause 300 to 700,000 deaths a year. And in the US alone, they're estimating that this year, 50 million people are going to get infected by influenza virus. So that's about 1 in 6 people in the US. And of those 50 million, they expect to be about 80,000 deaths. And so that's pretty significant, especially because we have an influenza virus vaccine, but the problem is it's not always the most effective. And the reason for that is because influenza virus, it can change its genetic code relatively quickly and dramatically. And this can cause the virus to then break out, and it can change from what's actually seen in the vaccine. And so that's part of the problem with the influenza virus vaccine and why people are working hard at trying to develop new vaccines. However, another strategy is to figure out how the body even responds to the virus in the first place, to try to understand better how we can fight it off and create better therapeutics, such as vaccines, for instance, or also treatments like different drugs. And so that's why we study NOD1 and NOD2 and trying to determine what they're doing in response to influenza virus.
So my hypothesis is that NOD1 and NOD2 can recognize and help control or kill influenza virus. And so a clever way to test this in the lab is that we have cells that have NOD1 and NOD2 and then cells that don't have NOD1 and NOD2. So we can infect these cells with the virus, and if NOD1 and NOD2 are important for controlling influenza infection, then with the cells that don't have it, we should see the virus grow more. And so what I did a few days ago is I infected cells that have NOD1 and NOD2, or they don't have them, and then I collected the virus from those cells, and I took that virus and infected a new layer of cells to do a plaque assay. So a plaque assay is an experiment in which you try to quantify the amount of virus that you have in a sample. And what you do is you infect a new set of cells with the virus, and these are cells that haven't been infected before. You let that sit and incubate for a period of time so that the virus can get into the cells, and then you take that off, and then you add what we call an agar overlay. So agar is this really cool component that's made by seaweed, actually, and we use it a lot in biology. And when we mix agar with some liquid food for the cells, which we call media, and then we put this on top of the cells and let it harden. And so you'll have a well like this. You'll have a virus that'll infect in this area, and this area. And it kills these cells, and it ends up leaving this clearing of where the cells are. So if you didn't have the agar overlay, it would just kill all of the cells, and you wouldn't be able to get these plaques that form. So that's what these are called, they're plaques. And so today what I'm going to do is develop this plaque assay that I did, so we can actually see the plaques and see the cells that are still alive and then the clearings where the cells are dead. And what we're hoping to see is that in the cells that have NOD1 and NOD2, we're hoping that we see less virus than in those that don't have NOD1 and NOD2. So I'm taking off all of these agar overlays, and so now on the plate, it's just cells that are on the bottom there. All right, so now I'm gonna add this cool purple solution. And this stains the cells, and it has what's called crystal violet in it. And crystal violet is just a stain or a dye for the cells. And it also has some ethanol in it, which fixes the cells to the plate so they don't come off. Okay, so let's get the dye to go all over the wells. Okay, and we'll let those sit for a few minutes so that it stains the cells really well. All right, so let's see if our hypothesis is correct. Now I'm going to develop these. I'm just going to take the dye off and then rinse them all with some water. So immediately you can see the plaques that formed. So the plaques are all those little clearings there. And each plaque represents where one virus first infected a cell, killed it, and then killed the surrounding cells. So the virus will get into the new cells that are surrounding it. All right, so I'll do the other plate. And it looks like the hypothesis is very correct. See this one, you see the plaques over here, these are completely clear because the virus has killed all of the cells because there was so much virus in that sample. So look at them on the white background here. So these are the cells that have NOD1 and NOD2, and these are the cells that don't have NOD1 and NOD2. So if we just compare these wells to these wells, you see no virus in these wells, lots of virus in these wells. So what we see is that when the cells don't have NOD1 and NOD2, influenza virus can grow really well. But when NOD1 and NOD2 are present, the virus isn't able to grow as well. So our hypothesis is correct, but now we want to know why this is happening. And so that's the next step. So on to more experimenting.